Anshul, Jack, thanks for two great presentations. So this was a really fun session. There's lots of different questions. I'm sure the audience will have questions. Let me just sort of get it started. And then I've also got the questions that you guys fed me that you're interested in asking each other. Let me just start with, this morning we had a session on interpretability in physics. And this afternoon we're doing interpretability in biology. And it seems like one of the key differences is that in physics, we usually have this very strong regularizer, which is like theory and math. And because of that, especially historically, we could learn from quite small sample size. And even perhaps many modern experiments in physics still have what you might think of as modest sample size, or they know how, as we saw in one of the talks this morning, they know how to put the sample size exactly in the small region of space that they're interested in doing the modeling. Now, it seems to me this is a key difference between biology as a natural science. So you guys have very complex phenomena. You probably don't have as strong theory. My guess is there's maybe a little stronger theory in genetics, perhaps, than there is in brain science at this stage of the game. But you're grappling with high dimensional spaces, modest amounts of samples. Now, it seemed like Anshul somehow had more data than I would have anticipated, which was nice. Jack, you still seem to be suffering, despite the fact that your data sets are large. You're still suffering in the lower sample size regime, where you can barely fit some of these complex models. I was just wondering how you could talk about the difference you think in the challenges of interpretability in your sciences, in biological sciences, say, in contrast to physical sciences, which I realize you're not physicists, but in this sort of absence of strong theory where you're more data-driven, what are the sort of challenges this is creating for you?
Anshul, you want to go first?
You can go ahead. I can go next.
Oh, you think I have an opinion about this? I kind of alluded to the difference between like classical physics and astrophysics in my talk, and I think that's related. You know, if you in classical physics where everything kind of sums together, and you can do things. There may be issues like linking across dramatically different measurement scales, but within a measurement scale, you can kind of make a coherent model. Yeah, that's a great place to be. And I think for neuroscience, we're more like astrophysics, where we don't even know what questions to ask in a lot of cases. And the way, if you kind of look at the history of astrophysics, well, you start with some, let's just look at the planets first. Like, let's figure out orbit. Okay, are there 5 classes of stars? It's like it's a very kind of course defined kind of enterprise, and I think that's the situation for neuroscience as well. The problem is our system is very, very non-stationary. So dealing with a very nonlinear, non-stationary system where almost any experiment you could do is going to give you a result that's going to differ from the natural world, it just ends up being a really hard problem. So I don't really have any great insights about this. I did find it interesting, however, that the very first talk today by Michel was basically he was towing the same line I thought. He was basically saying, I'm really data limited, although it wasn't clear to me how he's data limited compared to me, but he was arguing, I'm very data limited, and in this data limited regime, the deep learning is not that useful. And basically, you can hijack parts of it to help your GLM, but basically you're just going to be doing the general linear model. And I feel the same way for our data.
Anshul, you want to take a stab at it, or?
Yeah, so let me start with the data piece, and then I'll try to address the connection between like the physics strategy of modeling data versus the machine learning one. So we have a lot of data sets, but individually each of our data sets are actually quite small. So like the genome is very large, it's 3 billion positions. So it might seem like that's a lot of observations, but actually these, for example, these protein DNA binding events I was showing, most of them have about 2,000 to 5,000 to maximum 20,000 positions where they actually bind the genome. So it's actually there's a lot of background DNA where nothing happens. So there's contrastive negative data, but the actual positive data where things are happening is pretty small. Lucky for us, I think the actual function we're trying to model is not hyper complicated. So we have small neural networks, And linear models have been doing very well for a long time. Biophysics has dominated this area for a very long time, when it was much more difficult to actually measure all of these molecular events in vivo, in cells, in the genome. And so I think what's happening is actually we're going the other way around, which is, like I said, there have been beautiful biophysics models that encapsulate how proteins bind naked DNA, and they can make tremendous predictions, right? The problem is when people start moving from those models in these nice isolated systems to what's happening in the cell, which is there's tons of other stuff happening. They're trying to build models bottom up, right? So they're trying to build insulated models of proteins binding naked DNA, and then they add cooperativity, and then they add other components in the system, and very soon the physics gets very complicated, right? And it's very hard to model it explicitly. So we sort of taking the opposite approach now. Let's just take a black box, fit a neural net, and then we'll try to marginalize out the physics. Because like, if you look at individual components in that, you would hope that if the network can accurately predict these signals, right, it must have learned something, right? Some physics in there. And that's actually what we observe. So for example, if you take our models, which are trained on in vivo data, where all kinds of stuff is happening, right? There's multiple proteins, they are cooperating, there's kinetics, there's chromatin, there's concentration effects. The model has learned some version of that. Now, if we take that model and we literally take synthetic sequences, small sequences, and we just marginalize our over large number of backgrounds, just to consider the effect of that small little sequence, the predictions of the neural network correlate ridiculously well with like actual in vitro measurements of binding energies of proteins to DNA, which is fascinating. Like, I mean, it's encoded in the data. It's just some part of it, right? And you just have to find a good way to marginalize it out. Luckily, our inputs are very discrete. They're simple sequences, so marginalizing is not hard. It's just 4 letters. We can do all these kinds of tricks to do very simply marginalized signals. So, but what's I think interesting and exciting is I think we can start building bridges between the physics world, like the bottom-up approach to modeling versus this sort of more top-down approach, right? And somewhere in the middle, these things will meet and hopefully maybe build a bridge, right? Completely end-to-end. So that's my hope. And I think we're starting to see early signs of that, at least in protein-DNA interactions, protein-RNA interactions, and protein-protein as well.
Anshul, I think I heard you say that your intrinsic dimensionality is modest.
Yes.
For the problem, and that the phenomena you're trying to learn are reasonably simple and learnable, even with simpler methods. Jack, how many voxels do you have? And you also have a time dimension. And then, of course, presumably there's a very nonlinear phenomena that you're trying to learn. So this is one of the key differences, I guess, between the kind of problem Anshul's working on and the kind of problem you're working on.
Right.
What's the dimensionality of the voxel space? And even that's kind of coarse.
Well, I mean, yeah, in my case, I'm like where Anshul would have been if he's trying to solve this problem in 1995, right? We have an MRI machine. There's 80 billion neurons. There's 20 billion neurons in the cerebral cortex, and we have 100,000 voxels. So that gives you the dimensionality direction from 20 billion, to 100,000. There's millions of neurons in a voxel. And then they have this nonlinear coupling to the metabolism, which is what we're measuring. So the whole system is extremely reduced. In fact, you could argue it's amazing that you can get anything out of that. The measurement is so bad. But so we're already, when we're taking our measurement, we're already projecting down into a very low dimensional subspace relative to what's really there. And the dimensionality of our data just depends on our machine, basically.
And if I understand it, Anshul, you were able to do feature attribution in order to select down to an even smaller dimensional space that was important. But Jack, what you're having to do is some sort of PCA clustering kind of reduction, because you're not willing to throw away things.
Right, we don't know what's relevant.
Right.
So Anshul knows what's relevant and he's looking for something, and we have no idea what we're looking for.
Gotcha. So that's interesting. So Anshul, you can do experiments. Whereas, Jack, you're kind of working on the engine while it's running, right? And you have very coarse external measurements for the most part, right?
Yeah, we can make predictions. And usually, actually, in my case, the models that we fit in the lab, generally, since we're almost always in my lab, I would say 90% of the time, we're collecting data under some very rich naturalistic conditions. And the question is, does the model we build account for something somebody did under a simpler system? And that's usually the case. Because of course, if you build a more complicated model, it includes the simple cases as sort of special cases of what it can do.
And Anshul, the experiments you do are critical to the statistics that you need for what you're doing, right? If you couldn't run the experiments, I assume you wouldn't have enough data.
Yeah, so there are two types of, actually there are three types of experiments we have. We're, again, very lucky to be in genetics because there is the observational data that I just showed you. You can just look at these molecular profiles. There is natural perturbational experiments, which is genetics doing its thing in the human population, right? So we actually get mutations all over the place. There are millions of them all over the place. And so we can leverage that. And they are at different frequencies, different effect sizes. And then with CRISPR, now we can actually make artificial perturbations too. So we can fill in the gaps. Natural variation samples sort of extreme ends of the distribution of perturbations. And now the cool thing is to figure out how to use CRISPR screens effectively to fill in the gap. So now if you take neural networks and you train them not just on observational data, they're already doing pretty well. But if you couple them with actual perturbational information, they will completely, I think, get to where they need to get.
And Jack, just to close this thought out, like the astronomers, you're in a more observational setting where you can have them drive some visual toy system, but otherwise you don't really get to probe and modify the brain and the neurons.
Well, there are, as the same for Anshul, he has genetic mutations. We have brain lesions. I mean, there's tons of brain lesion data out there. And, over the past 20 years, the neurology community has done a great job of building up these databases that show, statistically, like, if you have a certain spectrum of behavioral deficits, what part of the brain is lesioned associated with that. The problem you have is that the assays, the behavioral assays, are usually pretty simple and low dimensional. And a lot of times people have brain disorders that they don't recognize or can't diagnose for a long time. For example, just take one example, Alzheimer's disease is oftentimes first noticed by the person who has it because they can't drive anymore. They can't navigate in a city that they know well, which is one of the reasons we set up this navigation experiment. Whereas if you go into the clinic, that won't be detected because that's not part of the clinical assay. So there are these brain lesions. I just wanted to mention one thing that I forgot to mention during the talk. Weirdly, there was a bunch of discussion yesterday about causality. We all like to think about causality. Neurophysiologists almost never think about causality. And the reason is it's kind of pointless. Most brain lesions, they don't have any specific effects. They just make you kind of suck slightly at everything. And that's because, you've got a lesion of some small region in a network that's highly interconnected. And so there are all these roots around that brain area that can kind of compensate for it. And so unless you get a lesion in a hub or some central, core area that all the information is flowing through, it doesn't have very big effects. So it ends up being this very complicated kind of network science problem that we can't really solve right now.
I want to go back to what Anshul said about you have the deep learning high level, you have the biophysics model at the cellular level. Can you speculate how you will bridge the gap? Does deep learning distillation will help you, and with what constraints? If you want to do a distillation from deep learning model, which is very successful, what would be the first step? What inspiration you will take from these biophysics models to do that step?
Yeah, it's a great question. So I can tell you sort of what we're currently planning to do and what we're hoping to move towards. The in vitro experiments are phenomenal, and you can really traverse the entire space of sequence complexity in sort of a isolated system, right? But even in that case, imagine you're trying to model sequences of size, even 40, right? Just 40 bases. You couldn't possibly generate a library of all possible sequences. Now, what we find is a neural network that's trained on in vivo data, like I showed you, like in cells in the genome. Once you marginalize out the component you're interested in, right, you can recalibrate it to an in vitro experiment that measures the same quantity you're trying to marginalize out. And it can be a pretty small experiment. It can be several thousand sequences. And once you've done that, you've calibrated the neural network to now be able to predict these actual biophysical quantities. And now you can generate millions and millions of examples, right? Because you can traverse the entire sequence space in silico. And then you can fit complicated physics models because now you have a lot more data. You can traverse the entire space much more effectively. And so there's going to be an interesting, I think, feedback back and forth where you collect a bunch of in vivo data. You're restricted to the regime of the genome, what you see, right? So your sequence coverage is pretty low. You can use in vitro data to fill in the gaps, to recalibrate. But then you can take those same neural networks and use them as generators to generate a lot more data to sort of, again, active learn the best experiments to perform next. And so to me, the neural network is not just like a black box, predictor. It's a component in an interesting feedback loop where we can now use it to design experiments, all kinds of experiments, and bring links between different experimental technologies as well. So I think that's pretty exciting, and I think it's going to power a lot of improvements in biophysical models too, which have been suffering due to being stuck in isolated systems and not having enough data to scale up as you add components piece by piece.
Have a success story already, or somebody follow this approach?
Can you repeat that? Sorry.
You have a success story already, or somebody did it for this approach?
Yeah, we've been doing this very simple. We've done this for individual, the same kind of experiments I was showing you today, protein DNA binding, but we've been able to link up in vitro and in vivo experiments, and it's really exciting. And we've been able to do this for proteins all the way from yeast to humans to very complicated systems, and it seems to be holding. So I think it's the first sign that there is signal there. There's biophysical signal in the neural networks. We still don't know exactly how to model cooperativity and all of that stuff, but it clearly provides a new framework to be able to generate lots of data in silico by coupling in vivo, in vitro experiments with neural networks to fill in the gap. So hopefully lots more exciting things to come.
Great.
Jack, I wanted to ask you a bit more about the not having enough data, because you said it, I think, in some different ways. One of them is, so basically, I want to know, do we not have enough data, or do we not have the data of the right kind, or do we not have the right representation of the data? So, which seem to be quite different things, right? Some of the things that you said about the dimensionality, like fourth dimensionality, which kind of makes us lose all information about many relevant brain oscillations, seem to indicate that we don't have the data of the right type, but might also indicate that we don't have the right representation. So maybe there are certain type of representations that are temporally based that we just don't even know about. So which type of so all types?
All of them.
Exactly.
We don't have all of them.
So do you think kind of machine learning techniques can help us make headways along this second and representation discovery?
Oh, the representation, sure, yeah. Fundamentally, we don't have enough space, right? We don't have enough spatial resolution because either in neuroscience right now, you can either get incredibly high spatial resolution, a really limited range field of coverage, or coarse representation over a large field of coverage. But of course, what you want is both. You want a large field of coverage with a lot high resolution. We don't have that. You can't get good time. If you want functional signals with good time resolution, you need to do neurophysiology in either extracellular or intracellular. If you do calcium imaging, which is a standard method used if you want to record from, say, 1,000 neurons, you also don't have any resolution over a second, better than a second, because the calcium indicators are very slow. MRI is very slow. So we have time problems. Besides just the time resolution, we have time scale problems. If the system is nonlinear and adapting, you need to record from it for a long period of time to have it transit through its different states. And nobody does that. And then we have, I would say, an environment problem. where, you know, an individual experiment will be under one local environment, but nobody has the capacity to probe the brain under a wide variety of different environmental conditions, which you need to do to characterize this nonlinear system. So that's all measurement. If you know, once you don't measure it, it's just not in there and it's just gone forever and you're just screwed. Based on what you have, I agree you should be able to use machine learning to get at those issues. I would say up to now, there have been no cases that I can think. I just want to make sure I'm really saying something that's true. There's no cases that I'm aware of or that I can remember, my poor memory skills, where somebody has used machine learning to discover a new representation that wasn't already kind of expected to be there in the brain somewhere. But I think that should be doable. And the obvious way to do it would be to do something like we have done, where, you know, you take a large number of feature spaces that essentially instantiate every hypothesis about representation that anybody's ever had. And then in parallel with that, you train a network in the same task and then take features out of that and, you know, adjudicate between those two. It should be doable. It's just nobody's gotten to that stage yet.
And I guess that could be a problem of explanation, which is maybe in some cases the neural nets have learned some representation that's novel, but of course we don't have a way of pulling it out of the neural net yet.
Right.
Yeah.
But the first step on that is just to show that there's variance accounted for by the neural net that's not accounted for by your models. And then after that, you know, you've got to parcel that variance out and try to attribute it, the same as Anshul has to do, right? What is causing this? What is it representing? What information is there?
I mean, I kind of focus on your story about the use of the neural network that's already trained, the Alex neural network. So it seems that there is here a phenomenon of some kind of a foundational learning, like that there's transfer learning. You do have lots of tasks.
Yeah.
You may not have enough data for each of them separately, but they have so much in common that the neural network that you develop now allows you to do some kind of few-shot learning, that you get a foundational model that captures some commonalities between many type of tasks. And after these have been captured, although we don't explicitly can describe the representation, this representation allows us to do some few-shot learning and save on samples. I mean, that's.
Are you talking about in the brain or in the network or both?
Yeah, no, I'm not trying to understand the brain. I'm talking about the network.
The network, yeah.
Yeah, but that's kind of my interpretation to your story about how you can use the network that has been trained on much larger image data, and now based on this you can do get along with much less data than you.
I absolutely agree. You can view this whole thing as kind of some transfer learning paradigm. And, you know, recently, I'm sure you guys pay attention to this, you know, Google and OpenMind and OpenAI, there you go, has, you know, they're training networks that play like 100 video games, right? And so you think, okay, well, a network.
There was an OpenMind.
What? Is that not OpenMind? I can't remember.
There's an OpenMind that is in cognitive science.
No, OpenAI. Oh, sorry, I'm senile. OpenAI, right. You know, if you have a network that plays 100 video games, you think, well, okay, that's got to represent a lot of features, unless it's just, you know, a sum of 100 networks, which presumably it's not. It's got to represent a lot of features that are of general interest and are probably represented in our brains as well, because the environment, the statistical constraints of the environment are far larger than people give it credit for, right? We're all living in the same environment, and the reason we see the same things is because it's an incredibly strong forcing function. The trouble is the networks that are trained to generalize are so huge that you can't do feature selection from them to project onto a limited data set. So again, we end up getting stuck again by the fact that our neuroscience data are so limited. If I have, you know, 1,000 features to project on my neuroscience data, I can probably solve that problem. But if I have 3 billion features to project on my neuroscience data, that's just, I'm never going to be able to do that. So that's the kind of problem we have.
I want to go back to this data size question that we've now been talking about for 10 minutes. I mean, you showed a graph, Jack, where clearly you don't have enough data to have the accuracy. You're not asymptoting in terms of accuracy.
Right.
And I'm guessing that that means when it comes to interpreting what the model has learned, to explanation, mapping it onto voxels, and all those sorts of things, that's not trustworthy either, right? I mean, if you have an asymptoted in terms of accurate prediction, then presumably your explanation is also kind of twitchy and not necessarily reliable. Or do you think that in fact it's reasonable despite not being up on the asymptote?
No, I completely agree with you, but one of the pathologies of science is that everybody ignores that problem. And I'm sorry, I got to go nonlinear myself here. So I had a paper once that I sent into a journal, and I said, here's our predictions. We're predicting 20% of the variance, and here's our model. And it came back from the journal, rejected, where the reviewer said, you're only predicting 20% of the variance. Why should I believe anything you say? This is ridiculous. Reject this paper. And I wrote back to the editor and explained, no one in the history of this journal has ever reported predictions before. Never, not once. There's not one paper your journal has ever published reported predictions. Only statistical significance. These are not, what is necessary but not sufficient. But the other, you have no idea what the other predictions for the other papers are. They could be 3%, 4%, and I'm like beating the heck out of them. And yet you're publishing papers all the time. Why are you victimizing me? And the editor said, that's a very good argument. Your paper's still rejected. So what we did is this, you want to see dysfunction. We removed all of the prediction from the paper. We resubmitted it as a new publication, same journal, it was published.
Dumb down the paper. Dumb down the paper.
Dumbed down the paper. We removed information from the paper and it became palatable to the reviewers and they published it. And I view that as a very seriously messed up thing. You should never be preferring a paper with less information, right? That's just bad.
And we have a very similar problem when doing machine learning in health sciences. So exactly the same sort of thing.
When you send in papers?
Yeah, exactly. But Anshul, I want to ask you a question. So you don't seem to be as data poor. Do you believe that you're up on the asymptote for the kinds of questions you're trying to answer right now?
I think for the quantities we are able to measure, we are doing pretty well. The problem for us is there are lots of unknowns, and most of the molecular events in the cell are affected by everything else happening in there. So we're just modeling like proteins binding DNA right now as a function of sequence. So it's one small component, right? But luckily, what's happening is the technology is allowing now folks to measure multiple modalities simultaneously in single cells. That's huge. We can get spatial information now about how cells are next to each other, which is huge. So I would say that our models are keeping up with the kinds of data we're able to get. But are we solving biology? To whatever extent we can, and we are mostly bottlenecked by what we can measure, right? A big bottleneck used to be perturbation data, but now, as I mentioned, with population scale sequencing and with really high throughput CRISPR screens, in the next 5-10 years, that's not going to be a big bottleneck either.
And you showed some beautiful graphs, whereas you varied the separation. You got more or less binding. If you were to just throw away the data, repeat the experiment again, the same sample size, would you get essentially the same graphs back? Is it that repeatable and robust at this point?
Yes, actually, in the paper, we in fact use different technology to measure binding, which is much lower resolution, and we end up seeing the exact same results. So yeah, these findings we're seeing are actually remarkably robust. And actually, I wanted to say a few words at some point about robustness of models and uncertainty. And yeah, so I can talk a little bit about that as well at some point.
Do it now.
Okay.
We're already talking about it.
So we were using these kinds of feature attribution methods and so forth for a while, for several years, and we were facing very similar problems to what people have reported in other domains like the imaging world and so forth, with extreme instability. You run the model twice, even the same data, and the attributions look completely different. And we realized, at least in our case, two things fixed it really, really well. One was naturally putting constraints on what we are modeling. So I showed you how we are modeling sequence mapping to profiles at single base resolution. The previous generation versions of these were binary labels. So we would take the signal and we would coarse grain them and make binary labels on the sequence, saying like this chunk of sequence is bound, that chunk of sequence is not bound, right? So we had a simple binary classification task. And there's a ton of summarization of information there from a sequence to a single scalar. And there are too many solutions to get to that scalar. And neural network picks a random one, right? Through pooling operations and so forth. As soon as we relaxed that and we decided to not summarize the data and just predict it as it is, it's noisy, but at single base resolution, that direct mapping from sequence to profiles, base to base, gave us enough constraint that it dramatically stabilized the models. We had to initially build feature attribution priors and whatnot, all kinds of machinery to stabilize the models. All of that is unrequired anymore. It just works. The second thing we realized is our attributions are very noisy. We were not seeing the nice, beautiful words I showed you today. And we realized all of that is coming from experimental bias. So what we decided to do is start thinking like statisticians, with respect to the outputs, and think like machine learning people in terms of mapping inputs to outputs. So we basically built a boatload of covariates into the model itself. So the model doesn't just try to predict profiles from sequence, but also includes a whole bunch of potential biases. The implicit regression of biases out of the signal simultaneously as the model is learning, it's beautiful. Like I didn't have time to show you this, but it is remarkable what the model learns after you give it information about what are potential experimental assay biases in the data. And the neural network does a much better job than any previous model in regressing out those biases as well. So two things fix the problem.
How are those biases entered into the model? It's not obvious to me.
So we just added as an additional modality. In fact, we can learn the bias from the data itself. So a simple way we learn the bias is we have these profiles across the entire genome, and there are only 20,000 actual events, which we can call quite reliably. If we fit a sequence model to the rest of the genome, right, the remaining billions of base pairs, the only thing you see there is bias, because there is no actual functional signal that's recruiting the proteins at those positions. So there's a huge amount of data to learn a sequence model that just predicts enzyme bias. And we can do this on an experiment by experiment basis for free. We take that model and we predict bias in the actual bound regions. We just add it as an additional modality, and now we have two paths the neural network uses, right? Directly going from sequence, and the other is this bias. So the bias regresses out as much of the signal as possible, and the sequence module only learns the residual. And the residual is beautiful. Like you can really infer latent patterns which you just can't see in the data, and it cleans up everything. So those are two things that really helped us improve bias modeling and then better objective functions.
Ben, can I ask a quick question before?
Yeah.
Or were you going to ask him a question about what he was just talking about? Go ahead. I'll ask later.
So like, how do you deal with the gene-gene polymorphism? Do you have that already for gene-gene interactions and also dynamic, right? Do you have a generalization to dynamic and nonlinear interactions? Yeah,
so we're doing two things now to make that happen. One is the receptive field of our models is still several kilobases. We're now building chromosome scale models, so we can actually, the entire sequence of the chromosome or the genome is seen for any event, to predict any event at any position, right? So now you can really start seeing complex interactions between loci, not just within loci, which I showed you. You can start learning those. And we are training directly on population data, not reference genomes, actual population genetics data. So we have actual variation going into the models, and the models can see how variation interacts. And lastly, we are modeling a lot of data at single cell resolution, which gives us a lot more information about variation, not just at the genetic level, but also at the cellular level. So those things are starting to help us. It's still a while away, but those are our strategies we're currently moving towards to learn interactions across the genome and other kinds of variation beyond, across single cells, for example.
Anshul, a lot of the great examples you gave today were kind of point mutations, or at least very local mutations. And what we would all like to see eventually is these methods being used to better understand, say, autism, which you mentioned today, or schizophrenia, an even more serious problem. And those are really complicated neurological deficits that are, probably either have multiple causes or are caused by the interaction of a lot of different genetic, problems.
Yeah.
So can you talk a little bit about how you're going to approach those problems? It wasn't really clear to me from your talk today with autism, and what you think the prospects are that machine learning might be useful for making progress on those diseases.
Yeah, so I think there are two important questions here. So one is, what we're exactly modeling. Right now, we're modeling what we call the cis context, just the contribution of sequence, which is only a small portion of any phenotype you see, right? How do we model the trans environment? That is everything else that the cell is reacting to that's not encoded in the sequence, the environmental response and the environmental stimulus. So the idea is to build larger neural networks that are hierarchical. They have a cis component and an entire trans component, right? So there's all other work going on in our lab as well as elsewhere, where we're modeling the trans components, signaling pathways, all of these aspects, right? And then how they interact with the cis component. And that allow us to model any cell state, right? It's not now one model for every assay and every cell type. It is one model for a cell, which has a cis component and a trans component. And those two components together define any cell in any state. So that's one component, which is like basically you have an entirely different sort of framework for trans effects, right? And the second thing you said is, and that requires you to look at the genome as a whole entity, right? So genome scale models. So I think cis-trans plus genome scale will take us to the next level. We can then model things like massive inversions, transversions, copy number effects, like much more complex variation that scales over bigger regions and has big impacts, in a networked fashion across the entire genome, right? And then you also have to account for the trans components, and together, I think that will be the next wave, basically. Both are happening simultaneously, but those waves are not connected yet. And when they do, I think we will reach sort of even better models of cellular response in disease.
Thanks.
Anshul, quick question before we wrap up here. I was struck by the fact that you had to create new methods of feature attribution, as opposed to using some of the existing off-the-shelf methods. Was that a computational issue, or is it because the off-the-shelf methods weren't really giving you what you needed? One of the reasons I asked is there's been a lot of discussion in the hallways here during breaks about existing feature attribution methods and whether they're good or bad. And I was kind of struck by the fact that you had to create your own.
Yeah, I mean, when we say create our own, they are very minor tweaks of existing methods, right? We kind of found the loopholes or at least potential problems. For example, gradient-based methods have issues with saturation, right? Like they don't propagate when neurons are saturated. And so we use finite difference approaches. Eventually, somebody else, like another team showed that our heuristic approach is like an approximation of these Sharpe scores, right? We hadn't even thought of that. Like we just did something heuristic that made sense to us, and it worked really well. And eventually, it was shown to have some interesting relationship to better understood theoretical properties of these things. At the same time, they're all empirical. We always evaluate them. Since we are largely applied, we evaluate them in terms of how they actually perform on huge numbers of data sets. So we have that, thankfully, capacity and amount of data that we can test them very thoroughly. And eventually the proof of the pudding is if we make a prediction and we can actually go test it out in a real experiment, then we don't care how it happened, right? If the attribution method is slightly unstable, but it still does a decent job at giving us hypotheses, we're okay with it, right? It may not have the theoretical guarantees, but it's okay for us for discovery. So, and like I said, somehow our domain, the data we apply these methods to, a lot of these attribution methods with slight tweaks end up really working extremely well. So it may be the simplicity of the inputs that we have, the discrete nature, and the other constraints that we have from the measurements, and the implicit causality from sequence to all of these events. Maybe the combination of those three things just makes small tweaks actually work pretty well. And some of our optimizations are just based on the architectures. We have very simple architectures, so we can leverage some of the properties, and we can just speed things up a lot. So it's all little tweaks. It's not like dramatically new methods.
And then Jack, I'm going to give you the last word here. Now, you were able to use sort of standard techniques often, it looked like TSNet, TCNets and PCA and things like that. But you were guided by an overall very complex system architecture that I guess is ultimately guided by what's already known about brain connectivity. What are the kinds of things, and this is one of the questions you had asked about, what are the kinds of things that you're finding lacking in the methods that you're using that you would really want, help in people trying to sort of push these things in directions that would make them more useful for the modeling you're doing, and explanation you're doing?
Well, I mean, right now, I think there is still a real limitation in applying from the point of view of machine learning, and applying these deep networks to brain data, especially in the regime of limited data. So, nobody who builds deep networks worries about limited data because they're applying it to problems where you basically have infinite data. So who cares, right? And so all of the optimization algorithms, all of the way the cost functions are implemented, all the evaluation algorithms are all they don't worry about data capacity. And there's this whole regime of extremely limited data that basically just nobody goes there. But that's our regime. So, ideally, what we would like is to dispense with all of these feature spaces, just use a neural network to fit to the data set we have, and then be able to interrogate the neural network, the fit network, and figure out what is represented, right? That's ultimately what we want to know. And frankly, I have no bias one way or the other on what algorithm gets used to do that. But again, as I mentioned when I was talking about just using the 3-layer networks to fit neural data, these networks are very data hungry, and we just don't have those data. So that's I think probably from the point of view of like what's lacking in machine learning, that's the main problem we have.
Gotcha. So a kind of deep learning that can be sort of ultra regularized to work with less data and not make the model too simple. It still has to be a deep model.
If it has to be ultra regularized, yeah. Yeah, I mean, that's the problem, right? Is what's the noise?
Introducing prior knowledge, and that's what we could see with the physics. That's there's so much prior knowledge, and that allows you. I mean, this is a basic trade-off of
exactly
machine learning 101.
Exactly. Yeah.
Okay. Hey, I want to thank everybody for sticking around. Thanks especially to Anshul and Jack. I thought this was a great session. Really lots of fun. So thanks for giving us the presentation and staying for the panel.
Thank you.
So we'll be back tomorrow at 9:30.
Thanks a lot, Anshul. Bye.
Bye. Thanks a lot. Talk to you soon.